Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about the membrane. Outside of the cell membrane, we're going to talk about 5 different things that can be out there. So, to understand what's outside of the cell membrane, we have to understand we're talking about different types of cells. So, outside of one type of cell, you can have what is called the interstitial fluid. Outside of a different type of cell, you'll have what is called the blood plasma. So as you guessed it, the erythrocyte, also known as the RBC or red blood cell, it is suspended in solution. The solution is blood plasma. So that is technically outside of the cell membrane. Number 3, the cerebral spinal fluid. A neuronal cell, the membrane of it is surrounded by cerebral spinal fluid. So as I stated in the beginning, different cells will be bathed in different types of solutions: interstitial fluid, blood plasma, or cerebral spinal fluid, depending on the cell type. Number 4 is cellular secretions. So outside of the cell, you can see cellular secretions. And number 5 is the ECM, extracellular matrix. So that's what we're going to spend most of the time talking about today is outside of the cell membrane, some cells, you'll have a matrix of sugars and proteins all interconnected. We're going to call that the extracellular matrix. The extracellular matrix is composed of sugars and proteins linked together. They're called glycoproteins. Also out there, outside of the cell membrane, are other carbohydrates that are secreted by the cell. So in the parentheses in this image below, all of that is the extracellular matrix. The extracellular matrix can be hard, or it can be jelly-like, or it can be fluid-like. The most abundant sugar protein in animal cells is collagen. So out there in the matrix, the most abundant sugar protein is collagen, which is a protein. We've talked about this protein in Unit 1. Collagen accounts for 40% of the total proteins in the human body. Previously, I showed you this image about the skin. I said, I have very young skin. Young skin has very good collagen and good elastin. These are 2 protein fibers that are in the dermis of the skin. As you age, as your skin gets wrinkled, if you smoke, if you do drugs, if you sunbathe, the collagen and elastin will deteriorate. This will create wrinkles. In the picture to the left, that is a picture of the matrix. So again, outside of some cells, you'll see the matrix. The matrix is nothing but proteins and sugars all interconnected together. So in that picture to the left, we're looking at the matrix. And in that matrix, you see the most abundant protein is collagen. So let's talk about where collagen can be found. And to do that, we're going to talk about 3 types of cartilage. We have hyaline cartilage, we have elastin cartilage, and we have fibrocartilage. The hyaline cartilage is the one that we will discuss. The hyaline cartilage is in blue in this image. Everywhere where you see blue. So let's look at the femur. The femur is attached to the acetabulum of the pelvic bone. We call that the ball and socket. The ball is covered with hyaline cartilage. This hyaline cartilage is nothing but a cell laying in a bed called the lacunae, and outside of the bed is the matrix. The matrix is heavily filled with collagen, and proteoglycans. So let's talk about the covering of that ball that asserts into the socket of the hip bone. All three types of cartilage are going to contain chondrocytes. Hyaline cartilage contain it, fibrocartilage contain it, and elastic cartilage contains it. That's the cell. That's the cell that makes that cartilage. That cell is encased in a small cavity called the lacunae. It's like a little bed that the cell rests in. Outside of that is the secretion, the extracellular matrix, which is secreted. So the extracellular matrix surrounds the bed, and it is composed of a jelly-like ground substance. So if it is in a bone, it won't be a jelly-like ground substance. It's going to be a hard ground substance that'll be high in concentration of calcium. But since this is cartilage, it's like a jelly-like substance. It's going to be, the matrix is gelatinous. It's not as firm as it would be in a bone. The only fiber that you will find in the ground substance out in the matrix is going to be collagen. So in this picture, I show you the cell, the chondrocyte. I show you the bed, the lacunae. And outside, I have it labeled ground substance. In that ground substance, you'll see collagen. This slide shows another image of the hyaline cartilage. I have two bones here. Let's say this is the humerus and how it attaches to the ulna. The ulna and the humerus are separated by fluid called the synovial fluid. The end of the bone has the hyaline cartilage. It's in light blue. So what I'm saying is the bone has its own cell type, osteocyte. It's in its own bed, and it has a extracellular matrix that is very rigid. But this light blue portion of the bone is called hyaline cartilage. In its matrix, it's going to be jelly-like. These fibers are embedded in a network of proteoglycans. So collagen is embedded in a proteoglycan complex. So right outside of the cell, you're going to have a bunch of proteins and a bunch of sugars all linked together. It gives structural integrity to the tissue. Let's take a closer look at just the proteoglycan. It is composed of a polysaccharide core, and it has branches that are called proteoglycan molecules. Each proteoglycan molecule consists of a core of protein and carbohydrates that are attached to it. So basically, if you look at this structure, it is nothing but sugars and proteins. These sugar proteins are attached in some cases to another sugar protein. This other sugar protein is called fibronectin. Fibronectin is attached directly to an integral protein that we're going to call integrin. Integrin is membrane spanning, means it goes from the ECF, the outside of the cell, all the way to the inside of the cell. The proteoglycans are also involved in cell recognition. So we know that the proteoglycans are involved in attachment. They help the cell stay attached to the extracellular matrix. It also is involved in recognition. So every cell in your body has a specific molecular fingerprint. My cells will have a different molecular fingerprint. So if we look at this image, we have that oval shaped blue circle thing that is embedded in our membrane. Attached to it are 3 hexagonal shaped sugars. So that is somebody's molecular fingerprint. A different patient might have 4 of those hexagonal green sugars. Another patient might have 2. Another patient might have 15. So every cell in their body has that same molecular fingerprint. The fingerprint is created through the proteoglycans. So proteoglycans are involved in cell coat, which is recognition. It helps to determine blood typing, and it is involved in immunity. If I inject you with Neisseria gonorrhoeae, your immune system is not going to identify it as self because you don't have these 3 hexagonal shaped, no, the bacteria doesn't have these 3 hexagonal shaped sugars attached. So therefore your immune system will try to destroy the Neisseria bacterial infection. Now we're no longer going to talk about what's outside of the membrane. Let's talk about the membrane itself. The membrane is going to separate the inside of the cell from the outside of the cell. It also plays a role in cellular activity. Now let's look at the composition of the membrane itself. The membrane is composed of lipids and proteins. The lipids are further separated into phospholipids, glycolipids, and cholesterols. The phospholipids make up the majority of the lipid content of the cell membrane, about 75%. The glycolipids make up 5%, and the cholesterols make up 20%. This slide shows you how it is evenly dispersed within this membrane. Let's specifically talk about the phospholipids. These phospholipids, we mentioned this in Unit 1, they have 2 sides. They have a polar side and a nonpolar side. The polar side is attracted to water. So we call the phospholipid amphipathic because it has 2 sides. The head is attracted to water, it's hydrophilic. The tail repels water, so it's hydrophobic. Why is the head attracted to water? It is attracted because the glycerol portion has a negatively charged phosphate group that was added to it, which gives it a charge. So now the slightly positive hydrogen from water will attach to the head. B is glycolipids. The glycolipids represent 5% of the lipids in the cell membrane. The glycolipids also serve as recognition particles. So the proteoglycans act as a molecular fingerprint, and the glycolipids act as a molecular fingerprint. C is cholesterol. So the nonpolar steroid-based cholesterol is embedded in the nonpolar portion of the membrane. This is how the membrane should look, the one on the left. A lot of the tails are unsaturated. Look to the right. If all of the tails were saturated, the membrane would be very viscous. So let's go back to the one on the left. All of these bent tails mean that the tail is unsaturated. Remember, every bent portion that we see, we need to add 2 hydrogen to straighten the tail back up. So if all of the tails are saturated, that would not be a good thing, because now the membrane would be too viscous, too non-pliable, non-flexible. You want the membrane to be flexible. So let's look at how increasing your cholesterol intake can actually harm you. So if you eat stuff like beef brains and chicken liver, and you eat a lot of it, you're going to increase your cholesterol. You're only supposed to take between 200 and 300 milligrams of cholesterol a day. Let's say you increase it. So if you increase the animal membrane's cholesterol, it's going to make the membrane more solid at moderate temperature. So if your cholesterol content of your cell membrane is supposed to have 4 cholesterols, your membrane is very flexible. If you double that to 8, then the membrane is going to be more rigid. Let's take this example and make it more relatable. Let's say it is an endothelial cell. That is a cell that makes up an artery and a vein. Let's say we're talking about an artery, and you have the endothelial cell, and instead of having 4 cholesterol, you have 8. The endothelial cell's membrane is going to be more viscous, more stiff. If it is stiff, then when the blood flows through it, it's not as pliable. So this is going to increase your blood pressure. That is not a good thing. So cholesterol is going to make the membrane stiff because it reduces the movement of the phospholipids. The phospholipids are constantly moving side to side. We call it lateral drifting. If you start embedding cholesterol molecules in the interior of the cell membrane, it's going to stop the phospholipids from moving. Therefore, it's going to make the membrane stiffen up. If the membrane stiffens up, you can develop what is called arteriosclerosis. Arteriosclerosis is 2 things: clogging of the artery by these plaques, or making that blood vessel too stiff. So we're only talking about making it stiff by adding cholesterol to the actual membrane itself. The second thing cholesterol can do is it can add fluidity at low temperature. All right? It can hinder solidification. So let's say there's a fish, an Arctic fish. You often wonder why this Arctic fish can be in very, very extreme environments and not turn solid. Well, if it takes and adds cholesterol to its membrane at low temperature, the cholesterol is going to hinder solidification. Remember, these phospholipids are constantly moving in the membrane, constantly moving, about 10 million a second. As the fish dives, as the water gets extremely cold, the phospholipids would tend to slow down. If they slow down slow enough, the membrane will become solid. That's why the fish can add the concentration of cholesterol, which hinders solidification. So this cholesterol at low temperature is going to disrupt the packing of the phospholipids. The last thing that cholesterol would do is it will disrupt the movement of ions through the cell membrane. Let's look at the movement of these molecules, phospholipids versus proteins. They can both move freely within the membrane. We call it lateral drifting. Now, a rare occurrence is flip-flop. So molecules typically don't flip-flop, but they typically will laterally drift very frequently. So the phospholipids will laterally drift about 10 million per second. Extremely fast. A lot of them will do it. Flip-flop is very rare. So this means one phospholipid that is facing the interior of the cell, 1 per month, can flip to the exterior portion of the cell. Now we're going to talk about integral proteins versus peripheral proteins. The integral proteins are membrane spanning and the peripheral are not. They're just going to be located on one side. The integral protein penetrates the hydrophobic interior of the bilayer. That gold circle blob that penetrates that membrane to the right, notice that it only penetrates the interior. It doesn't go all the way through to the top phospholipids. So that is a characteristic of an integral protein. It penetrates the hydrophobic interior. The gold blob to the left penetrates the interior and goes all the way out. So it is membrane spanning. We call that a transmembrane integral protein. So both of them are integral proteins. Most integral proteins are involved in transport. They're going to either carry molecules across or they're going to form a channel to allow molecules to flow through the pore. These proteins can also serve as enzymes and they can also serve as receptors. So all of these are things that integral proteins can do. Now let's look at peripheral proteins. They are not membrane spanning. They are not embedded. So they don't pierce the hydrophobic interior. They're going to be attached to an integral protein. Some peripheral proteins act as enzymes. Some are motor proteins involved in mechanical functions. And some link cells together like a rivet. So think of the cells of the skin. The cells of the skin are anchored to one another through these rivets that we're going to call peripheral proteins. They help hold adjacent cells together. Both lipids and proteins collectively lead to the description of the membrane. You heard this in high school. They always said the membrane is a fluid mosaic model. Why do they say that? Fluid means that something is always moving. The phospholipids are constantly moving, 10 million per second. The proteins are moving. So that is where they get the term fluid. Mosaic means, this distribution of cholesterols and glycolipids and proteins and peripheral proteins versus integral protein. All of that is just randomly dispersed in the membrane. So therefore they call it the fluid mosaic model. This is how the fluid mosaic model appears. Notice the randomness of all these different molecules, phospholipids, cholesterols, and proteins. Now we're going to look at the function of these actual membrane proteins. We have several functions we're going to look at. The first one is attachment to the ECM. The first 10 minutes of our talk, that's what we looked at. The extracellular matrix that was composed of collagen and proteoglycans. So the protein that is embedded in the membrane helps the cell stay attached to the matrix. The second thing that the proteins that are embedded in the membrane will do is they are involved in cell signaling. This is how the cell communicates with other cells. One cell on one side of the body is going to produce a chemical, release the chemical, and it travels through the bloodstream and stimulates a cell on the other side of the body to do some response. That is called cell signaling. The third function of a membrane protein is enzymatic activity. So some of these proteins will take, let's say, a sugar like maltose and break it down into glucose and glucose. The fourth thing a protein that is embedded in the membrane will do is it will be a transport protein. It can form a channel or it can form a carrier and move molecules in and out of the cell. The fifth thing that these membrane proteins can do is intercellular joining. I talked about the rivet. So therefore, if you look at E, the cells of the skin, you got two cells anchored together by a rivet. And the last one is recognition. Again, every cell in your body has a specific, unique molecular fingerprint. All of these are created through these membrane proteins.